Okay. All right, so you already know who I am, and we're going to just dive right in since we're already way behind. So today I'm going to talk to y'all about plant health and plant disease, disease management, plant disease symptoms and signs and examples, plant pathogens and disease examples specifically, and disease diagnostics and exercises. And finally, some resources and other info. I'll probably leave that for home reading just because we are running so short on time. All right, so what is plant health? Plant health, in a nutshell, is when a plant is free of disease, stress, or injury and can reach its full potential in terms of yield or other production measures. This may be flowering. It just really depends on what your scenario is. So there's a ton of different definitions of plant health in textbooks. Every textbook will have its own, but generally all of them will focus on this. All right, so why is it important? Well, there's a lot of reasons, but really focusing on maximizing plant health can help maintain security and stability of our food production. This is really important with supply chain stability. We've had some hiccups with that in the past couple of years. Also, safe food to eat. That's another really big deal for a stable society. Also, we strive to maintain profitability of crop production for farmers and landowners, and this includes direct cost of disease management to farmers, so like fuel and fungicide and things like that, as well as yield loss, the money they're not making, both in quantity and quality. For example, if corn is a low quality, the farmer's not going to get as good of a price at the grain elevator as they would if it were, in premium condition. And things like fungal pathogens in particular do have a pretty significant role in the quality of not only grains like corn or wheat, but also oilseed crops like soybeans. All right, so what exactly is a plant disease? So when we're talking conversationally, just kind of throwing things around, generally people kind of refer to any problem as a disease, right? But when you talk to a plant pathologist, we're talking about everything on the yellow side of the spectrum here, so over here. And all these issues are what we would consider biotic, or the root cause is from a living thing, like viruses, bacteria, fungi, oomycetes, parasitic plants, nematodes, insects, and other herbivores as well. So primarily, as pathologists, we just focus on the ones in this green circle. Now, there are a lot of abiotic or non-living problems that affect plants, and in all honesty, the vast majority of the time you have a problem with a plant, it's going to be on the blue side. So things like chemical burns, soil pH issues, nutrient imbalances, low oxygen in the soil, in terms of smothering the roots, drought, and so on. All right, so pretty much all plant parts can be affected by plant diseases, roots, stems, leaves, fruit. And we categorize diseases in kind of two main groups, soil-borne and foliar pathogens. And those are primarily ones that we see with the initial infection on the above ground tissues versus the initial infection occurs in the soil. And it also kind of refers to where the pathogen resides. So things like fungal root rots or root knot nematodes, those live in the soil and infect the plant through the root, versus something like alternaria leaf spot that can live in old plant debris laying on top of the ground, or it can be seed-borne. And the primary means of infection is through above ground tissues. So again, those main types of pathogens we're going to talk about today are fungi, and that's in bold because it's really important. It's the biggest group we're going to talk about: bacteria, viruses, and nematodes. All right, so y'all should have seen the disease triangle in your notebooks. If not, you need to go back and look. But the disease triangle refers to the 3 points or 3 factors we need to have present at the same time in the same place for disease to occur. So you need a susceptible plant host, you need a virulent pathogen that can cause a disease on that host in particular, and then a favorable environment for that disease, or for the disease to progress. So basically the pathogen needs to be in an environment where it can be happy and reproduce and thrive. So this is a really important concept to understand, because through disease management efforts, our goal is to disrupt the disease triangle, preferably at 1 or more points at a time. I like to say try to do 2, if not all 3. because then you're increasing your likelihood of success. So again, it's a helpful tool to visualize opportunities for disease management. Okay, so now we've talked a little bit about what we need to have present for a disease event to occur. I have a very generalized disease cycle here. So all plant pathogens have a disease cycle. This isn't quite the same as a life cycle, it's just close. So this is the path that it takes for the infection event and manifestation of symptoms and things like that. So we'll start up here at the top. Overwintering is kind of like a rest period for the pathogen. I like to call it overseasoning here in Texas because we don't really have winter. This can occur in plant material or an insect vector, you know, and by plant material, I meant like living plant material or old dead plant debris in the soil itself, in the absence of plant debris even. So there's a lot of ways that it can overseason and have that kind of downtime period. And for some pathogens, that might be the heat of summer. Other pathogens, that might be the coldest part of the winter. For some, it might be really dry conditions. For some, it might be really wet conditions. It really varies pathogen to pathogen. All right, so if that pathogen comes in contact with a plant host and favorable conditions occur for that pathogen to start its infection cycle, then we have infection, which is when the pathogen actually gains entry to the plant and overcomes plant defenses. Now, if it was seed-borne or it was already present in a cutting or something like that, this will look a little different. But for general purposes, we do have an infection step. So next, the pathogen starts to cause disease symptoms in the plant as it continues to grow and colonize throughout the plant tissues. And then it reproduces and spreads. And in many cases, we'll kind of start over at that overseasoning stage. Now, some pathogens do have secondary cycles. I said cycle, but it can be cycles within a single season. So you can go straight from reproducing to finding a new plant host. And then over and over and over again, like you can have dozens of secondary cycles within a single season in some cases. All right, so on to disease management. So, and because we're really short on time today, Dale, please hold your questions to the end, and then I will gladly stay after and answer as many questions as you have. So, all right, so disease management. The management strategies that we really focus on are resistant or immune plants, so non-host plants, avoidance through cultivation techniques, exclusion, protection, eradication, and therapy. And we're going to talk more in depth about these. So these first four are focused on prevention. Notice they're first, meaning they're extra important. You should pay attention. And the last two are treatment. We all end up in this boat sometimes when you do have a problem and you do have to, apply some treatment principles, but we do prefer to focus on prevention. I'm a big fan of prevention over dealing with a problem. All right, so the first one we'll talk about is, again, the most important in terms of it's the most bang for your buck, it's the easiest, and that's resistance and immunity. So this is when we employ plant genetics and traits to naturally resist the pathogens. So this is a really classic example of pepper that is resistant to Phytophthora blight, right here on the left. And then on the right, we have one that is susceptible. That is the only difference. They were not managed differently. One was not exposed to more pathogen than the other. It's been identical other than their genetics. So this is a really great example of just how simple and easy and just how good a result it can deliver. So there's two types of plant resistance. There's direct, in which the plant directly prevents disease using genetics. So there's some things that are coded in the plant that respond directly to a specific pathogen. And it's able to recognize that pathogen and just be like, hey, we don't do that here. You need to back it on up. And then the plant is able to, through a series of chemical reactions, sometimes it's the leaf, sometimes, the chemical response will actually inhibit the pathogen. It really just greatly varies. But directly, you can think of it as like a direct interaction. Indirect is more like just kind of happenstance things that are also beneficial in terms of resisting disease. So these are plant traits like a thick waxy cuticle that prevents pathogens from gaining entry. So thick waxy cuticle, for example, would have other purposes as well, like preventing sun scald or reducing heat of the day transpiration and water loss and things like that. So it's kind of a multi-purpose trait, and it just so happens that a really thick waxy cuticle can help resist penetration from germinating spores in some cases. So direct and indirect. And that's a little advanced for y'all, but I do like to kind of give you a sampling, if you will. Again, this is your best option with the least effort, and it's the most cost effective. The drawback is we don't have a resistant variety of every crop that we grow for every pathogen that's out there. But this is a significant area of research and effort with crop breeders. So, new ones are out every year. All right, avoidance is when you avoid disease development by making the environment unfavorable for the pathogen using cultivation techniques like adjusting your irrigation method and timing, using mulch to reduce splash and the need for watering as frequently, tillage to help kind of prompt decomposition of some plant material and also remove plant material from the surface for foliar diseases, for example. Different or altered planting sites to less favorable areas for disease or areas with better drainage, for example. Different planting times to avoid a favorable season for the pathogen. So, for example, let's say we have fungus X that really, really, really loves the hot part of the summer. So if you can plant your crop early and get your crop harvested before the peak heat of the summer, then you've just avoided a problem. So that would be an example of different planting times to avoid the peak season for the pathogen. And lastly, use a variety adapted to your area to avoid stress that will result in weak plants. Just like people, if you're all weak and stressed and run down, you're just not going to have a good immune response. And plants don't really have an immune system like people do, but they do have a response system. So if it's already just kind of struggling just to live, it's not going to do well. So in a nutshell, it's manipulating the environment, which is also known as cultural disease management methods. All right, exclusion is exactly what it sounds like. You're using methods to keep the pathogen out, using only healthy plants, don't bring in sick, sad little plants, buy certified plant materials, especially for y'all with things like citrus. Be very careful with, buying citrus that's not from a reputable source in terms of the quarantine that y'all have going on through most of the coastal bend now and the valley as well. So maintain clean equipment. You're preventing moving things from spot to spot. And this, we talk about that a lot with commercial growers, but even within your yard, I wouldn't, just willy-nilly go from one garden plot and then loan my tiller out to my neighbor and let them use it in their garden plot without hosing it down really well first. And include infrastructure to maintain a clean growing area. That's just helpful to make sure you actually follow through. I know if I have to go on a wild goose chase for, the hose and whatever it is I need, if I have everything like there and ready all the time, I'm much more likely to follow through. The limitation to this is you can't control everything. Something will get in, it's inevitable, but we're really just practicing risk mitigation and trying to stack the deck in our favor. All right, so protection is utilizing a barrier of some kind to protect a plant from the pathogen. This can be a chemical barrier like a fungicide, biological barrier. There are a lot of biologics out now. They've gotten very popular the past couple of years, and they can work in a couple of different ways. Some are by directly colonizing the surface of the plant. Some are making metabolites that antagonize the pathogen. So there's a variety of activities that are sold with the biologic selections now. Lastly, a physical barrier, something as simple as a raised bed. If you have a variety of tomato that you just absolutely love, but it's susceptible to everything under the sun, I would recommend, maybe considering putting it in like a plant or a raised bed or something, just because you are reducing the likelihood of having problems, especially if you have a history of, say, Fusarium wilt in your garden plot. Well, doing a pot or a raised bed is a simple way to just avoid the problem and protect your plant from infection. However, protection is not that helpful if you already have a significant problem. Eradication is killing or getting rid of the pathogen by removing or destroying diseased plant debris through sanitation or roguing. I prefer the term sanitation, but you might come across roguing in some older texts. Some people used to do fumigation in the area of planting paired with solarization. Nowadays, it's more biofumigation like mustards and things like that with solarization. Solarization is a process where you use clear plastic, which you can see on that picture on the right, and you put some moisture on the soil, put the clear plastic down, and just let it sit for weeks and let it get up to around 100, and I think it's 122 degrees, but don't quote me on that. I have the fact sheet if y'all are interested. So basically, you're killing off as much of the pathogen as you can, and this is really helpful with like soil-borne pathogens or really heavy cases of like foliar pathogens, and you really want to jumpstart that decomposition process. A lot of people do like to follow this up with like kind of reseeding their soil with some beneficial microbes, maybe some mycorrhizae or something. So that's always, you know, kind of a bonus that you can add on afterward. So this is again for after a pathogen is present. And there's a little reminder about biofumigating with brassicas. All right, so other therapies are helping the plant to heal using systemic chemicals paired with pruning. I always recommend these together, never one at a time, just because you're far more likely to have success when you pair up methods. And with pruning, you want to prune out diseased, you know, infected materials as much as possible, and then do your systemic chemical application. Now, a lot of systemic chemicals are for pesticide applicator licensed individuals only. So you might have to get like an arborist out to take care of a tree or something like that. All right, so disease symptoms and signs. So symptoms are changes in the plant in response to a pathogen. Just like, for example, if you have a cold, your symptoms are fever or, you know, runny nose or whatever. That's your body's response to the virus, right? So same thing with plants. A symptom is the change in the plant in response to the pathogen. This can include wilting, yellowing, or chlorosis, tissue death, or necrosis, stunting, which is when it's too small and just generally unthrifty, and galls. Signs are direct evidence of the pathogen, like actual pathogen tissue. Typically, this will be like fungal tissue, sometimes bacterial ooze, but that's not as common. Okay, so we're going to do a little kind of work through here. So this is a cabbage leaf, and we have signs and symptoms both in this photo. So symptoms would include yellowing or chlorosis, tissue death or necrosis, and wilting. And we have signs here. You can see it better zoomed in here. This is just cropped from here. So signs are these little black flecks and this white tissue here. It's kind of like cottony. So that's actual fungal tissue. In this case, the little black specks that look kind of like coffee grounds are reproductive structures that make spores to then spread to new plants. And that's just a little reminder arrow for your notes. All right, so some really common symptoms I'm sure you've come across: yellowing and dead spots, leaf blight, leaf spot. Spots and blights are often going to be from fungi, bacteria, and viruses in that order in my experience. That doesn't necessarily mean it will always be true in every case ever, but that's just kind of the order of prevalence for my area and vegetables specifically. General yellowing through the whole plant and just kind of like unthriftiness can be nematodes, just a little clue there for y'all. But here's an example of a leaf spot. On the right, that's a tomato with alternaria leaf spot. On the left, that's actually a peanut with alternaria leaf spot. Now, these are two different species of alternaria fungus. And this is just a really good example of how even a closely related fungus like causes very different looking, yet kind of similar symptoms. So if we just verbally describe it, well, they both, you know, are round, they expand and merge into each other. Here you can see how that one's merged into each other. And here we had a couple individual spots that merge together, and same here. And we have that here as well. So we call that coalescing leaf spots. So they coalesce or merge together. We also have some kind of black coloration in the middle here. And if you look closely, there's a little bit there too. It's just far more distinct here. And that's actual, spores, just thousands and thousands of spores that individually are very microscopic, but, when they're in mass like this, you can kind of see them. So this is a good example of, you can have a really broad host range with certain fungi. Others are very specific. So as with everything in plant pathology, I'm sure I sound like a broken record, there's a lot of variability. Here's another example of a different type of chlorosis or yellowing and necrosis. Now notice the language stays pretty similar, but visually it's very different. In textbooks, we would describe this in more detail with more jargon, but for, kind of early learners, it's difficult to read about these things because the language can sound the same. So we have yellowing and necrosis. Well, we had yellowing here and necrosis as well. So, it's important to try to add additional descriptors like, yellowing along the veins, progressing to necrotic tissue along the veins that expands outward to encompass the whole leaf. Versus here, we would describe that as leaf spots enlarging into merged areas or coalescing into large areas of the leaf. So we start out with very kind of general terms, but we can get more specific even without using a ton of jargon. Okay, and there's just a little vocab tip. There will be little vocab boxes all along the way in the presentation. So some other common symptoms are cankers and diebacks. These are often fungal, sometimes bacterial. This is rose. I see this a ton on rose where we have a fungal dieback. I see a lot of phomostis dieback with this, and it's exactly what it sounds like. The canes just die back, and it gets further and further down. So, a lot of people think, oh, I could prune here. Well, that's not the case. You actually need to prune way down here because it's kind of like an iceberg. What you're seeing is not the extent of the infection. So I always recommend going at least 4 inches down and then pruning, and use clean clippers, for example. So again, I see this a lot with roses and ornamentals. All right, here's another example of tree canker and dieback. Here, it's infected the twig and made it back to the main branch. So in this case, you couldn't say, for example, just cut the twig off. You would have to go and remove the entire branch to try to reduce the inoculum there. Galls are tumor-like overgrowth on oftentimes roots. It can be above ground tissues as well, but I do see this a lot on roots. And it can be caused by fungi, bacteria, or nematodes. So I also see a lot of root and crown rots, especially in like seedlings and houseplants and succulents and things like that. The things that don't like to have wet feet. Here we have a healthy seedling with kind of creamy white roots. And here in the middle, we have a really, really sick little seedling. That's just a goner. If you look, the roots are really unthrifty. If you were to press into them with your thumbnail, they'd be really soft. And they're this dark chocolate brown color. So there's no saving this seedling. It has a really severe damping off. So this next panel right here, you can see that it's kind of in the middle. It's not that healthy creamy color, but it's also not a complete goner. So this is a really good demonstration of plant diseases that are not always all or nothing. There's a spectrum of severity, and a lot of factors influence that severity. Everything from how much inoculum was present when it infected the plant to the environmental conditions, and how vigorous the plant was to start with and things like that. So this is just a nice little reminder, like a visual reminder of that. Typically, this will be caused by a fungal complex, so multiple species of fungus, occasionally paired with nematodes, and occasionally bacteria, but more often than not, it's at least 1, if not 2, fungal organisms. All right, so plant pathogens and disease examples. All right, so first we're going to talk about bacteria. These are single-celled organisms These are everywhere in the environment, but only some are pathogenic to plants. Leaf spots, wilt, bacterial scorches, soft drops are all very common with bacterial diseases. Most that cause disease in plants will not make humans sick. However, if you're immune compromised, there's a really good little fact sheet from Iowa State that's at the link right there. Yeah, y'all should go to and read up on it. You know, it's just good knowledge to have. So, one other side note, some bacteria that are pathogenic to people can survive on plants like E. coli, Listeria, Salmonella, and others. Those are obviously something that's always of concern, particularly if you're working in like, you know, community gardens or eating your own produce or sharing produce. So there's a ton of really great online resources about, you know, produce safety from the home garden, especially if you're using like manure-based fertilizer. That's a major risk factor, in my opinion, if it's not aged manure. So, Kevin, I'm sure there's a ton of resources you can share with them. So hit up your county agent. All right, so these photos here are examples of bacterial wilt. Here on the, actually, we're going to start with the bottom panel here. This is actually a quick test way to determine if you have a bacterial wilt. If you have a plant that's all wilted and sad, and you wonder if it has bacterial wilt, go to the main stem, like kind of down by the crown, and then cut it with a clean razor blade or very sharp knife, put the stems back together and very slowly pull it apart. And if it's, oops, not too far. If it's really sticky and slimy and strings together like that for like a 1/4 inch or more, then that's a good sign you might have a bacterial wilt, because all that really goopy material is actually the exudate from the bacteria that it secreted while infecting the plant. Another method is to cut your stem. This is actually a tomato stem, and put it in a cup of water, and it will ribbon out like this into the cup of water. Now, this is a particularly severe case, so it's really visually obvious. You can get like one of those little black light flashlights like people use to find like pet messes in their house or something, or scorpions, for example, and shine it behind it, and it will really help see that. And I'm going to try to get my video to play. Hopefully it will let us. You know what? I'm just going to send the video files. Like, I'm going to post a link to them and send you all the link, because I don't think it'll let me do video. All right. So it's a really cool video too. It's a shame that I can't get it to work, but it is what it is. Okay. So with bacterial wilts, this is what they look like typically. You're going to have a really water-soaked and limp appearance, and it will be followed pretty quickly with plant death, or at least a significant portion of the plant will die quickly. In the early stages, it's most apparent midday when it gets really hot, and it will be really wilty despite watering, and that's because the vascular tissue is actually really damaged and actually physically clogged up as well. So here you can see a tomato stem that's been cut lengthwise, and this chocolatey kind of rotten looking area is actually bacterial wilt inside the stem. That's, as you can see, you know, just completely decimated the vascular tissue. This does spread easily by water splash and wounds, particularly insect feeding. So some prevention tips are prevent insect injury, reduce water splash, kind of prune things out, or increase your plant spacing to really let things dry out and reduce that canopy humidity. Cull infected plants, practice good sanitation. I don't like putting plants that are diseased back into compost piles. In theory, if you got your compost pile up to temperature perfectly every time and got every bit of it, you know, to the right temperature, then it would probably be all right. But for me, it's not worth like the risk in terms of you might get 2 cups of compost from it, you know, so I personally don't choose to do that. So also rotate your crops. If you have a problem with this, rotate for a minimum of 1 year, and that includes no volunteers or weeds. I know we all like little happy volunteers because they're like free plants, but just if you have a problem, don't keep your volunteers around. So I do see this most often in like cucurbits and that family, and tomatoes. All right, so citrus greening is an issue, so it's an area of interest for y'all. And it's caused by bacteria that infects the phloem of citrus and will eventually kill the tree. It's Liberibacter, and it's also known as Huang Long Bing or HLB. But most people here in the U.S. will either call it HLB or citrus greening. So it's vectored by a little insect called the Asian citrus Asian citrus psyllid, which is right here. It's itty bitty, very small. And it can be spread by people moving plant material like cuttings, even young budwood and leaves. So again, be really careful where you get your cuttings and things from. So I have a little kind of sketch thing here that I made in the computer. So this is going to walk you through how vector events happen. So here we have a happy little psyllid. Imagine this is beautifully drawn. And it has the bacteria on board. So it lands on an uninfected leaf and it starts feeding with its little mouth parts. And then it transmits that bacteria down into the phloem, where it then replicates over and over and over. And here, these little kind of cylinder globs, those are individual bacterial cells. This is a cross-section of the phloem tissue of a plant. So you can see these cells are actually physically clogging up the vascular tissue. And these also secrete toxins and just damage the plant internally. So there's a lot of really gnarly things going on. So that's just a little kind of peek into a little bit more advanced material for y'all. All right, so symptoms include discoloration of fruit, small fruit that are bitter and lopsided. That's a classic kind of red flag. Raised or thickened corky veins. So here's an example of that. And asymmetrical blotchy modeling of the leaves. So unlike a nutritional deficiency, which is usually going to be a pretty close mirror image on either side of the leaf, this is going to be asymmetrical if you can look at the two halves, like down the midrib. So it doesn't match up. Like all that modeling doesn't have a counterpart over here. So that's one of the most common symptoms here in Texas. That's kind of like a red flag. So if in doubt, always ask. You know, Kevin down there is wonderful. Also, Dr. Ong up in College Station is great. All right, so detection is a major thing, especially with the commercial industry. Currently, there's no cure for infected trees, so prevention of infection is key, and that involves removing infected trees so you don't have like a typhoid Mary situation where it's just sitting there pumping out inoculum to infect neighboring trees. There are some sprays for the vector, but these little guys can fly away. They kind of do that like leapfrog flying where they'll fly away and then rest and fly away and rest. So they can be really challenging to manage. Early detection and action to help limit the spread is super important. And removal of trees right away is very, very important because every day that they're out there is another day that they could be infecting other citrus trees. Areas where citrus is grown is of great concern. I can't even explain to y'all how much of a concern this is in terms of the billions of dollars in the industry that's been lost. So quarantine is a really important tool, and that includes efficient mapping, vigilant detection and scouting. And detection is really kind of the frontline thing that we have right now. So DNA screening is really sensitive, but you can get false negatives if you don't collect a good quality sample. Or let's say you collect from the right side, but the tree was only recently infected, so the left side is where the bacteria is proliferated and it's not fully systemic in the tree. So you can have issues like that. Dr. Femi Alabi in West Laco has also been doing quite a bit of work on citrus screening as well. And in fact, there's actually been a really successful program using dogs that are trained to recognize the scent of citrus greening to hit on trees for further testing. So that's kind of one of the newer advancements, and they have very specialized canines and handlers for that. All right, so this is an old map, but you can go to the citrus greening quarantine area website and look at the most recent map. Both plant material and the insect vector has been detected in shipments in these areas. Y'all are pretty much all in a quarantine area, so that's something to be aware of. And again, this is an outdated map. It's just a screenshot. So go check out the most up-to-date information. All right, so viruses are submicroscopic, and many are spread by vectors, which are often insects. Some can be spread by mechanical means like clippers or your hands, and some may even be seedborne. Yellowing, loss of vigor, and death of the plant in most cases occurs. Here's an example of a virus symptom. It has a very distinctive bullseye pattern. This is actually a peanut leaf with tomato spotted wilt virus, and here's a tomato with spotted wilt virus. So this can be a little confusing because we have a peanut leaf with tomato spotted wilt virus and a tomato with tomato spotted wilt virus. And I mean, the symptoms even look kind of vaguely similar in their pattern, but the tricky thing about viruses is that the way they're named is kind of unusual in that they are named for the host they're found on first, the symptom, and the word virus. So this was first found on tomato. It causes spotted wilt virus, or spotted wilt, and then virus. So kind of after this system was in place, we later realized in the pathology community that the same virus can actually infect a lot of different hosts. So tomato spotted wilt virus, for example, can infect a lot of different things, including tomatoes and peanuts. Another example is cucumber mosaic virus, infects most cucurbits as well as a ton of other plants. Tobacco ring spot virus or tobacco mosaic virus. That one's one of the oldest known viruses, and it infects many, many different plants. So if you're reading along in a seed catalog, let's say, and you see a tomato, it says it's resistant to tobacco mosaic virus. You're like, why do I care? It's, well, it's actually very important because it can get tobacco mosaic virus, for example. So that's how that system works. So hopefully that will eliminate any confusion for y'all. Some other symptoms are kind of malformed growth, like you can see here on the picture in the center. And I included this photo on the right because discoloration or subtle discoloration is like, I wouldn't even call this subtle, but like discoloration in general can be a little tricky to kind of spot. In passing, this might look like a nutritional issue, but if you look at it more closely and look at the pattern, for example, if you look at this leaf, it is not symmetrical at all. So to me, that would be a red flag that we're not dealing with a nutritional issue. Like this leaf right here would look more like a nutritional issue to me, though that would be extraordinarily severe. So if we had that much yellowing, I would be shocked that the plant would have leaves that well developed. So just kind of look for some of those clues, but particularly, especially starting out, the patterns of discoloration can be a really good kind of tattletale thing for viruses. So prevention tip, prevention through vector management, getting good clean stock. If you're, taking cuttings or something like that, tool sanitation, and co-affected plants. So a prime example is rosette virus. This has been documented in Texas now. It causes these symptoms like malformed leaves, discolored leaves. These are a little too red and they're mottled and yellowed as well. Also, note the thorns are deformed and there's way too many. And also sometimes you'll have kind of crowding between the nodes, so the distance will be way too short. And sometimes you'll even have, say, like, 30 malformed tiny flower buds where you should have one or two. So just general kind of growth misregulation is very common with this. And it is spread by mites. So mites feed on plants that are infected, get the virus in their mouth parts, and then go feed on fresh plants and infect them that way. You can also spread this by not sanitizing your tools between plants when you're pruning. So that's an example of mechanical spread. Physical disturbance of the plant tissues, like going from infected to non-infected and handling plants a lot and really getting sap on your hands quite a bit. Though the spread efficiency on that method is not nearly as high as like mites. These always result in plant decline and eventual death. So prevention tips, start with clean stock, space your plants so they don't touch, so those mites don't have just like an easy highway from plant to plant. Sanitize your tools, remove any wild multiflora roses, so y'all probably don't have a ton of those, and call those infested plants like as soon as possible because you don't want a typhoid Mary sitting there spreading to all your other roses. So one tip, and this is only good for viruses, it does nothing for bacteria or fungus, or nematodes. But for viruses, like if you're doing a lot of pruning or cutting flowers on roses, you can dip your shears in milk, and that denatures the virus protein. So the proteins in the milk actually pull apart the proteins in the virus proteins, because the virus is basically genetic material with proteins surrounding it in various shapes. So if that protein coat is damaged, then it can't function anymore. So that is a quick and easy way to sanitize your pruners between snips if you don't want to wait for, say, like Lysol to do its job or alcohol to do its job, because you can't just like spritz Lysol on it and then immediately cut. Like you need to wait. You know, if you read the instructions on the can, you've got to wait for, like, you know, whether it's Lysol or bleach or alcohol, you need to wait at least a couple minutes. So little fun fact tip there. All right, so tomato spotted wilt virus, we talked about this on the first virus slide. This is actually spread by thrips, and monitoring for those with sticky traps and insecticides applied as needed is a great way to manage those. And with this, oh, I forgot to discuss symptoms, sorry. So this is the spotted discoloration as described in the name. And we do have kind of some wilting symptoms, but notice these are much smaller than the other tomatoes back here. They're just generally looking very poor and unthrifty and discolored. And on the stems, you'll often see this kind of chocolatey brown, almost like pitting, sort of. And on the fruits, you have that really classic kind of bullseye or ring spot appearance. So with this, remove the infected plant material and weedy hosts in the area, because they can actually be a reservoir for the virus and the insect vector. So that's why weed control is really important. And use the resistant tomato varieties. That can be a really, really great tool. There are a handful out there, and they're excellent. I'm a big fan, so I always recommend those. All right, nematodes. So these are microscopic worms. They infect plants and feed on nutrients from the plant. Most are root feeders, and they infect through the root. They do have a special mouth part called a stylet that is unique to plant pathogenic nematodes. And here's a close-up of that. It's like a little straw, kind of, that they use to pierce the plant material. And here you can see some root knot nematode galls on carrots. All right, so nematode problems in Texas. They span many different types of nematodes. It depends on your soil type. The most common issue I see with homeowners would be like root knot nematode, which prefers sandy soil, but can do just fine in clay soil. Typically, root knot has a very wide host range, so it can be a little tricky to find a rotation crop for those. I do come across things like reniform nematode, but it's pretty rare. Though you guys are a lot sandier where you're at, so you might have a different experience with nematodes. So under the microscope here, oh, sorry, that should have animated first, but here's root knot on pecan roots. And this is a very susceptible variety of pecan, so you have these huge galls. And the galls are actually made up of the bodies of the female nematode and feeding cells, which are cells that have been kind of hijacked, and they become super large, kind of like a tumor. And they crank out tons of sugars and proteins and things for the nematode to eat. Now, the other part that makes up the gall is actually going to be the body of the female. And here's a really good shot of that right here, where you have an egg mass inside the female. So you can see individual eggs here, these little white ovals. So it's pretty crazy that galls are actually malformed cells and egg masses, and basically the dead body of female nematodes. So prevention tips are plant-resistant varieties, prevent infestations on spec transplants for any signs of root knot or anything like that. And crop rotation, like a 3-year crop rotation plan, can be really helpful on just preventing problems and preventing buildup, even if you already have a low level. All right, so fungi. This is the largest group that we have in terms of just the sheer numbers of samples that come into my lab. The good news is most fungal organisms out in the world are just decomposers. They're not pathogenic. They're just living their life, breaking down old dead plant material for the nutrient cycling for the ecosystem. Others are symbiotic, so they're actually helping plants, and y'all can read all about those. But I mean, I'm sure you guys are familiar with mycorrhizal fungi and the impacts it makes. All right, so this is also typically the group that we most often see, quite literally. It is highly diverse. both in its appearance of reproductive structures and also how those reproductive structures function. So some are easily visible with the naked eye, like these jelly ear fungi that are actually sporulation structures, which occurs right here, ranging from that to like bread mold, which has microscopic sporulation structures that look like this, where you have these little they almost look like brooms with spores that bud off the tip. So that's what that looks like under the microscope. So it's just a dense mat of these and some vegetative growth. Here's another good example. I'm sure you've all seen these. These are conchs. They have spores that form on the underside here. And these are another good example of something that can be a decomposer, but I do often see this as kind of a weak pathogen on trees that are already struggling. Okay, so back to fungal structures. The mycelium or hyphae is the cottony white substance that you see that looks kind of like threads or like pulled out cotton. This is the kind of body of the fungus, if you will. So this is the part that colonizes the substrate, whether that be dead plant material, a dead animal, it doesn't matter. It's whatever is colonizing it, or colonizing, that's your hyphae. So this is a sign. It's not always visible because sometimes it is deeply embedded in tissue. So just for perspective, this is where the mycelium, which is plural for individual hyphae, would be, like with a mushroom, it's in the ground. And then you have a mushroom that comes up out of the ground that you actually see, and that's the reproductive structure and what makes spores. So, all right, so onto spores. These are kind of like seeds. When they land on a suitable substrate and have favorable conditions, they'll germinate and colonize the substrate. In the case of plant diseases, it's going to be typically a plant material of some sort. So here we have a spore, starts to germinate, these little thread-like structures, and it'll continue to grow. And here is a kind of a side view of a slice of a leaf, and the blue dyed threaded-like structures here, that's fungal tissue colonizing this kind of tan background is the plant tissue. So that's like a microscope cross-section of fungal tissue embedded in plant tissue. So you can see how, when I say it deeply colonizes, it gets up in there. It's not just kind of loosely associated, it's in there. So here's an example of a spore that's landed on a leaf surface. The green cells here are the leaf surface, and it's actually had enough moisture and favorable temperatures, and it started germinating, and it's actually trying to gain access through the stomata, which is actually a pretty common way for spores to gain entry. Some spores have these specialized little structures where they can actually force their way down between the cells here, called macrosloria, and it's pretty cool. It uses really high pressure to literally like bust through the cells. So here's an example of one type of spore, kind of like teardrop shape, I guess. This is actually alternaria. It's the same thing that was on the tomato and the peanut earlier. And here's a shot of fusarium spores. Note, these look very different. Now, y'all don't have to remember these like specific names or anything. Just know that spores are incredibly diverse in their shape and color. And that's actually a diagnostic feature that I use to identify what we're dealing with. So what I do is I'll take a little scraping or a little pinch off of an infected material piece. I put it under the microscope and I look at it. And that I look at the shape, the color, the size, and everything, and I use reference materials and figure out what exactly we're dealing with. So it's a diagnostic feature. All right, so how do these spores spread, since they are the primary reproductive structure for most fungal pathogens that we'll deal with? Well, they can be spread by wind, water, splash, even people. We will start with something familiar. Here we have a mushroom, and on the underside we have gills. And if you took a cross section of that and looked at it under the microscope, you would see thousands and thousands, probably millions, of these little structures that make spores, and then the spores drop off from the gills and they get caught on the wind and then go find their new home. I actually have a video for this that I will send y'all a link to later tonight. So this right here is actually a gall or in this case, it's actually a fungal reproductive body on cedar. This is cedar apple rust on the cedar stage. And this actually is a kind of, we call them teliohorns, but just it's a sporulation structure. And it makes the same type of spores that this does right here. So thinking about, where the cedar apple rust sporulation structure is occurring, we're high up on the tree. If you think about it, what do you think the means of dispersal for these spores would be? Do you think it would be splashing water or wind? Well, if you think wind, you are correct. So the primary means of dispersal for this is wind. So, the structure of these reproductive structures, like the physiological makeup, as well as their location, can be a really big clue on how the spores disperse, which in turn can be informative on how we would take management efforts. So in this case right here, we have a little microscope cross-section of a leaf. Again, that's the kind of green right here, that's the leaf. And here we have these little structures that are poking up, kind of like sea urchin. And these are actually fungal reproductive structures. And here, that's what it looks like in cross-section if you were to slice it down the middle. It's kind of like a little cup with these little spikes, and it makes spores that kind of ooze out. So here I have that fungus in culture, actually, and you can see those little itty-bitty structures. They're kind of crowded up here, but back here you can see them individually. And this goop on top, that's actually spore ooze. So it's oozing out these spores on the top. So if we had a rain event, it wouldn't, be surprising to know that droplets of water would hit these, splash out, and actually spread the spores very efficiently through water splash. So those are both classic examples of how the reproductive structure has a role in how these things spread. And again, here's your little cheat sheet box. So here's the link to the mushroom spore video. We only have about half an hour left, so I'm going to press on, but y'all feel free to go take a look at it. It's super cool. Okay, so I see a lot of early blight in Texas. This is caused by alternaria, the fungus we've talked about a few times. It favors moderate to warm temps, high humidity. It overwinters in old crop debris and alternate nightshade hosts. It can be seed-borne. And note, when I say alternate nightshade hosts, that includes potato. So in areas like here, where we're far south and we have year-round production of various crops, there are extra risks. It's hard to break that disease cycle when it has a host available pretty much all the time. So unfortunately, it can be a little tricky to spot the early lesion spots, but later on, you can see that classic kind of ring appearance. And if you were to rub that, you get a little bit of like powdery black material in your hand, and that's the spores. So prevention and treatment, resistant varieties, minimize splashing, optimize that airflow to dry things out, remove infected older leaves to reduce the just sheer numbers of spores you have there. End of season sanitation, that includes those tempting little volunteers and alternate hosts. So anything in that Solanaceae family. You can always pair these things with fungicide applications. And if you cover your tomato production area with UV absorbing vinyl, we can reduce disease by about 50% because this actually needs UV to sporulate very well. So you're basically just kind of almost half neutering the fungus, if you will. So that can be a helpful tool for homeowners. And practice good rotation. It's always just good practice to have good crop rotation on a 2-year schedule for this disease. And again, there's a little reminder what the spores look like. And you can actually kind of sort of make out the chains of these spores with a really good hand lens. All right, powdery mildew is super common. It is light to pale gray. It'll look like this on a leaf. Sometimes you'll see little black specks, not always. Here's a close-up of those little black specks. Those are actually reproductive structures. This fungus has 2 types of reproductive structures, though. This is the sexual reproduction stage right here. These are called chlysosthesia. You don't have to worry about that. It's just a fun word. But it's filled with little sacs of spores. Now, the asexual reproduction is actually these little kind of beads. It almost looks like a clear pearl necklace, if you will. That's actually largely what's making up this white material on the top of the leaf. So These are individual spores that are beating off of the surface of the leaf. What else? Yeah, so these are visible with a really good hand lens. You can get by with an okay hand lens to see these Clystothecia, but different pathogen species do infect different plant species. This is very, very host specific. So like powdery mildew on roses will not infect your crepe myrtles. Powdery mildew on crepe myrtles will not infect your zucchini. So it's very host specific. It overwinters in old crop debris, so sanitation of all that old junk is really important. And also, I say related plant hosts nearby, but that I should have put volunteers. I shouldn't have put related, that's my mistake. So spores are blown in, and on crop residue, blown in is like one of the hardest things to challenge because you cannot stop the wind. Crop rotation is not very effective due to the long distance spore dispersal. So even if your neighbor 8 houses down has it, you could still technically get it. Seed treatment is not effective due to exposure times later in the season. So, even buying treated seed is not going to be protective for this disease. So again, resistance when available, sanitation of heavily infected materials, wait it out if you can, because hot, dry weather is really just an unhappy time for this pathogen. So it's going to really be inhibited. You can use fungicide. If you do, stay on schedule. That's super important. And you need to start early and be continuous with it. Some people say potassium bicarbonate, which is not quite the same thing as sodium bicarbonate, which is baking soda, but potassium bicarbonate can be effective. Some people say milk diluted 1 to 10 with water can be effective. I've never seen like published data on that in a scientific journal. That's not to say it won't work. If you see great results, let me know. All right, so fungal-like organisms. These are problematic in our area. I come across these all the time. These are also called oomycetes or water molds. These include downy mildew, pythium, phytophthora, which cause root rots and damping off. And they have a very unique measles spore dispersal. So here we have downy mildew on the underside of a spinach leaf. Here we have a classic root rot on some strawberries with like this really dark black rot. And if you were to tug on that, it would just slide right off the cortex of the root. And here's a classic shot of damping off before it even breaks the soil. All right, so some common things that I see with this here in Texas are Phytophthora blight of pepper. It survives in the soil and crop debris. So let's imagine this sad little pepper were to drop down, and if it got left there in the field, it could serve as a source of inoculum for next year. So policing this stuff is very important. In the future, if you have a history of a problem with this, replant with resistant varieties. If you don't, practice good fungicide use and sanitation of infected material, and be really, really vigilant. And also make sure you get clean seed. So never ever save seed from like sickly looking plants like this one. Key things on recognizing this, you have these chocolatey brown to black kind of necrotic areas on the stem that will start lower and work its way up. It'll also look almost like greasy, not necessarily wet, but kind of just like greasy. And then on the fruit, this is probably the most like recognizable thing, is you have this really greasy looking line of progression, and then you'll have heavy sporulation behind it. So that's kind of a very classic look for Phytophthora blight. All right, so one of my first samples ever actually was hydroponically grown grass. And, well, wheat. I should say wheat. So this is unfortunately a bad picture because it was my old cell phone, but here you can see all this fungal tissue, all this mycelium just like all over these roots. And they're not a healthy creamy white color. We have a lot of discoloration. So here, this is a shot of a little root that I plucked off and put under the microscope. This is around, I want to say 100 times magnification. And I noticed within the root, we have all these little balls everywhere. So I zoomed in to around 450 times magnification. These right here are plant cells, the long rectangle kind of like trapezoid looking things. Those are plant cells. And then these are actually the reproductive structures of the oomycete. I believe this was a Phanomyces, actually. So inside here, we have these little kind of blobby looking things, and those are actually zoospores. And zoospores actually behaves very differently from a typical spore. So we're going to see if I can make this work. Maybe. Fingers crossed, guys. Maybe. Okay, so this will be one of the things I send you in the link because it's not loading right. So I'll send you the zoospore release video. Basically, these zoospores actually swim around like little tadpoles. I hope this one might work. Yes. We'll give it a second because this is really, really cool and I really wanted y'all to see it. I'll let it buffer for a second. So those spores release from those little round structures and they swim around, like actively swim around. You can see here. So we have the sporangia. Those are the little round structures. They release from those and swim around through a thin film of water. And even in the soil, there's often a thin film of water in the little spaces between the soil particles. And these are microscopic, so they can swim around all through that. And this is under a microscope. They've just put a little droplet on a slide with a little root here. So actually, well, as you can read here, the zoospores actually follow nutrients that seep out from the root, particularly down here in the root tip area. That's where the highest concentration is. So in the water, we have all these sugars and hormones and great things that the zoospores can actually sense. And then they will actually swim toward the increasing strength of that, kind of like you can follow your nose to cookie smell. So once they get there, they insist, not like put your foot down like you insist something, but like insist, like ENCYST. So I call it set up shop. So we have all these zoospores swimming around, and then these, like this guy and this one, they set up shop. So they then start behaving more like a typical spore and will actually germinate a little hyphae down into the plant. So they're very different than, say, like a mushroom spore that just drifts down from the mushroom and lands where it lands and just, you know, germinates whenever conditions are right. So it has a much more active means of dispersal. And that's just one way of dispersal. There's a lot of other reproductive structures with ORCs, but that's the most like diagnostic, you know, in your face feature that makes them highly unique. So some diagnostics and exercises. We're going to run through these pretty quick. So accurate diagnosis is super important because incorrect management attempts just to make things way worse. You're wasting your time and your money. And fungicides only work on fungi or oomycetes, with the exception of copper. It is labeled to work on bacteria as a preventative only, but it does nothing for viruses or abiotic problems. So accurate diagnosis also lets you know if you need to focus on insect vector issues. And the first step in diagnosis is identifying your host plant. Next, you need to assess the problem, do your research and develop a suspect list and narrow the list of suspects and get help as needed. So this is just to give you all a look into what happens so you can better understand what information specialists like myself need. You're not going to be expected to, you know, just diagnose a plant right off the bat, you know, and honestly, we'd prefer you didn't. We just want you all to have a working knowledge so you know what information we need. Okay, so ID that host plant. Your plant owner, county agents, or other master gardeners that have been doing it a while are super helpful for this. Reputable online resources, taxonomic keys, and field guides. The main thing here is be careful of common names because they're not universal, especially regionally, so that can be a little tricky thing. And it's really important to know this because it automatically eliminates a lot of possibilities because not all diseases or not all pathogens can cause disease on all crops. Next, you want to assess the problem by examining the whole plant top to bottom and the surrounding area for any clues of anything that might have happened, especially with abiotic histories. Consider your site history and decide, again, if it's even a disease, is it an injury, a disorder, things like recent weather, particularly hail or old freeze damage. So don't only consider like last week, but like especially this past winter, we're still seeing calls coming in about, hey, what is this? And it's just freeze damage. Also consider if the soil been tested to determine nutrient status, to see if there's a deficiency there. Various chemical sprays, lightning, and I also asked about duration of symptoms. Has it been going on multiple seasons, or was it seemingly overnight? Typically, if it was seemingly overnight, it's going to be an abiotic problem. Diseases take time to really develop and get going. So how many plants are affected is another issue. Is it a single species or more? How is that area distributed? Usually if you have like hard lines or of dispersal, that's going to be a man-made thing. With pathogens, it's usually going to be more like a gradient. Doing the research and developing a suspect list is the next step. After evaluating all your symptoms, looking for signs can be a good clue. And then I use reference books, and there's some really great references online. There's also a lot of just utter trash online, so please be careful with where you go look. Use reputable resources. Most university distributed items are going to be peer reviewed a little bit, like we'll send it to some colleagues and they'll look at it before we publish it out for the public. But if it's just, some dude's website, just proceed with caution. And also consider what's common on this plant. If it looks like a duck and quacks like a duck, it's probably going to be a duck more often than not. All right, so that final step is always the most tricky. With this, you're pretty much always going to need a specialist. So there's actually a great series of books, though, called the Compendium series, and they should really pay me as much as I plug this for them. It's made by the APS Press, which is like the main publishing group from my field of study. And there's tons of different ones covering different crops. They are a little expensive. The digital copies are less expensive, but there's nothing wrong with like a group of people, buying, say, rose diseases and pests and keeping it for the Master Gardener group, or bean diseases or whatever your favorite crop is. So these also have wonderful color photos, descriptions of problems, not just diseases caused by biotic issues, but disorders, insect problems and things like that. So they have pictures, which are super helpful when you're just starting out. All right, so case studies. Let's say someone brings you some leaves of multiple species. They all have a rough leathery texture and they're widespread on lots of different plants of the same genus. And there's some yellowing. So the first thing you would do would be ask the individual what the host is or what the plants are. The second thing is thoroughly inspect the plant. Take note of all the symptoms. And okay, so there's your little answer box, your host ID, and assess your history. So not just the symptoms, but ask 20 questions of the person who brought it to you, like were there any sprays relatively recently? Is this somewhere that's had a lot of digging nearby? This was actually, I think 5 or 6 different species of citrus, or different varieties of citrus, I should say. And it's actually turned out to be scale insects and mites, like severe mites, which can cause this kind of yellowing and curling. Okay, case study 2. Someone brings you a dying plant, like the whole thing. It has severe defoliation, wilting. What would you first ask the person, and what would you first do to the plant? And this is what it looked like. Here's a relatively healthy plant and the sick plants that were brought to you. So first, you would ask the person, what is this plant? It is sesame. So what would you do to the plant? Well, we would take stock that, we have severe wilting, defoliation, it's stunted, a really poor pod set, really poor root development. There's some discoloration. It's hard to see on the ground there, but there was some discoloration along here. Here you can see a better shot in the lab. So you have discoloration on the outside of the stem. So another key place to look is inside the stem. So split those stems, look inside for discoloration. In this case, we actually had charcoal rot and rhizopsia rot, both. So we had 2 problems at the same time, so it was a little unique. Well, I shouldn't say it's unique. It was the first time I had come across that in my job, because this was the second sample I ever got. So that was pretty exciting. But charcoal rock causes kind of a grayish discoloration on the inside, so you can really kind of see that right there, which is where it gets its name. So inspect the plants and the surrounding area, top to bottom and inside. All right, so some home reading, because we're at 8:20. Here's some bonus slides. I'll go over this one real quick. So this is actually a gall on a tree here. This is on the trunk. This one was from the crown area of another tree. And these were caused by fungus. Now, in like forestry situations, woodworkers have actually discovered or determined that there's a way to, you know, cut and like polish this wood to where it's really attractive. So you have things like burl wood that people use for inlays and pins and all kinds of things like that. And it's actually a very expensive, very beautiful wood. So I like to include this because it shows that, you know, even if we have a plant that's, you know, not done well and even been killed from a pathogen, you know, there are some nice things that can come out of it. In this case, burl wood. All right, bacterial soft rots. These are not super common, but I do see them from time to time, particularly in onions. And this is going to be a late season and post-harvest issue. And bacterial soft rot pathogens actually have an enzyme that breaks down the cell walls in the plant material. And that's why it's soft rot and mushy. And one thing these have universally in common is they really stink. So anything that's mushy and really stinks, that's going to be a bacterial soft rot more often than not. Especially in onions, because that just smells like hot trash. It's so gross. The one thing to know about this is if you do have an issue with this, even like the tiniest spot, let's say on this carrot, it was just like this little spot, you still want to call that out because if you store it with other like healthy produce, it will contaminate it and ruin the whole batch. Key prevention tips: limit insect injury during the growing season, especially that later half of the season, and you know, anything that's even suspect post-harvest. So y'all can read that on your own. Oak wilt you can do on your own. Cotton root rot. We can talk about this real quick. I think we have the time. So with cotton root rot, it's a fungus that does typically thrive in high pH and high calcium soil, but it does just fine in like neutral pHs. So you know, when I say it thrives in high pH, that doesn't mean it can't live in areas that have a lower pH. As long as it's above around 6.5, 6.8, then it does fine. It's just not going to be, you know, at its maximum performance. The main thing you need for cotton root rot is really hot soil temperatures of over about 80 degrees. So with cotton root rot, it pretty much will kill the plant, usually fairly quickly. Often it'll be at least or minimum of like a week, but max of like 2 weeks to maybe a month for like a really large plant. Here you can kind of see an up close of the kind of almost like drought stress looking symptoms on this grape. But once the leaf dies, they stay attached. So that's kind of like a red flag. And also, obviously, these are irrigated grapes, so it shouldn't be having drought stress. So here's a shot. It's kind of not the best picture, but here's a shot of like peaches where you can see the leaf has stayed attached. And this does have extreme longevity in the soil, like at least 100 years that we know of. It survives with little modified hyphae that are kind of like little tiny pebbles or nuggets that are called sclerotia. So those little tough structures can live for a very long time in the soil. It is fairly widespread. It occurs through all the Southwest U.S. and down into Mexico. And it also does have a very wide host range. It's over like 1,000 plants that it can infect, including cotton and grapes and fruit trees and olive trees, vegetables, many ornamentals. And again, it has kind of a wilted, scorched appearance and rapid death, with the dead leaves staying on after the plant dies. There's a distribution map. And this is what it looks like if you dig up the roots. you have what looks, I kind of liken it to flattened yarn almost. This is a very heavy case where it's very heavily polished, but it really shows it nicely where it's kind of like tan yarn that's been squashed on it. It does make a spore mat, and it's usually kind of with like salmon to maybe a creamy color, and it'll emerge after a rain, and it usually is anywhere from like 3 to 6 inches in diameter. The unusual thing about this, though, is the spores seem to be sterile and don't have a known role in distribution. So as far as we know, it's primarily just distributed by, flooding events, washing, fungal material, various areas, especially within a field, moving just soil and earth, with like plowing and, even like moving fill dirt from one place to another. So there's some theories about like maybe animals being able to move it, but that's not ever been proven to my knowledge. This is really difficult to manage. Resistance is your best option. There's a list of resistant plants at the end of your handout, or not at the end of the handout, a link to the resistant plants is at the end of your handout. Commercial producers do have access to fungicides, but that's been really only in the past 10 years. And this has been an issue for over, I'd say upward of 150 years, like with agriculture in this state. As of now, it's cotton, olives, and grapes. I hear there are others coming, but I don't know what the timeline for that is. The only for sure means of diagnosis is digging up the roots right away while they're still fresh and sending them to a specialist for us to visually identify it. That's the only way we can detect it right now, and we do not have like a soil test for it or anything, just due to sampling limitations with that. All right, so there's a little slide on integrated pest management and an example of that y'all can go over as homework. Okay, so abiotic issues, I see those a lot. This is an example of herbicide injury on a citrus, actually. And plants do vary greatly in their susceptibility to various herbicides. Citrus is super, super sensitive to glyphosate, which is Roundup. So even just like, invisible to the naked eye droplets that might catch on a breeze can do this. So you'll have these kind of very defined edges on these spots, and they'll be kind of bleached white looking, and they don't grow. Like with a pathogen-based issue, these spots would enlarge over time, and you would have more and more and more spots more than likely. So that's one of the key differences is this will look like it shows up almost overnight, and then it just stays the same size. Unless you have enough spots on this leaf that the plant just gets stressed enough that it's just to completely drop the leaf. But even then, the individual spots will not grow. Okay, so insect issues. Dr. Irfan is no longer in Overton, I don't think, but I believe he's still in the A&M system. I need to get his current contact info, but Molly Keck in San Antonio, she's your closest one right now. Well, no. Holly, I think her name is Davis, down in Westlaco, the entomology person in Westlaco. She was hired on last year, so she would be your closest person probably. But Molly in San Antonio is really good too. These are just some basic ID things. I know just enough to be dangerous about insects, but beyond, stay on schedule with your spraying, whether it's soaps or insecticides, whatever you choose, just make sure you stay on schedule. Sooty mold is not really a plant parasitic fungus. It's actually just a fungus growing on the surface of the leaf, consuming the honeydew, which is basically poo, from the insects that are just feeding on the plant. So if you get your insect problem under control, the sooty mold self-resolves pretty soon. Some people do like to kind of blast that off with like a garden hose or something. And if it's really heavy like this, you might have a little defoliation, but it's not going to be permanent. All right, parasitic plants. I see a lot of mistletoe. It is a true parasite. It does like feed off of the tree and take nutrients from it, as well as dodder, which is like an orangey to sometimes a really pale yellow-green vine that doesn't have any visible leaves, but it can have these itty-bitty tiny like pinhead-sized flowers and fruits that actually make just tons of flower-fine seeds. And it's really difficult to control if it's already gone to seed. So try to get to that early if you have it. And here you can see a really great shot of the hostoria, which is a special little structure. It's like a sucker looking thing, but it's actually not. It attaches and has roots, basically, like modified roots that go into the host plant. So it looks like some weird alien thing, but it is actually a true flowering plant. So, and here's a cross section of what that looks like. So these, if it gets out of control, these can be really problematic, but especially on plants that are maybe like trees that are a little stressed already, then it can be a major problem and really hasten their decline. Mothballs on here, though they're not truly a parasitic plant, they're actually an epiphyte like an orchid. They're just living their life on there. And you can remember this by thinking about all the places you see mothballs, things like stop sign edges, on a barbed wire fence, on an electrical line, like there's no food there for them to eat. So they're not consuming anything. They're just living their life on it. But some people don't like the way these look. And so you can treat them with potassium bicarb. Baking soda is sodium bicarb. Technically it will work, but it's not going to be as effective. And on the parasites, focus on physical removal, though if it's this bad and the tree's already this stressed, there's no point. You may as well just take out the tree. All right, so resources are here. Shipping instructions for samples are there. And resource websites. And then there's my contact info. So this will all be in your handout, obviously. And here's a survey. If y'all could just do this survey and send it to Kevin. And Kevin, if you could facilitate that in addition to the survey that y'all do in-house, that would be amazing, because having a uniform series of questions for all the groups that I do is very helpful in my data to report for my boss. So if y'all like the class and want me to keep my job, please do the survey. And then lastly, I have a little home activity for fun, because I know deep down y'all are nerds like I am, or you would not be attending an evening class on a Monday evening to learn things. So you can do mushroom spore prints next time you'll see a mushroom out and about. And here's the instructions for that. So now we will open it up for questions.
I have a question. First of all, thank you for this. It's very informative. Loved it. Got a lot of information out of it. Can you talk a little bit about animal waste, whether it's bird waste, dog waste, those kind of, any type of animal waste going into plants, and what kind of disease it might cause, and what signs we can identify? Specifically problems with animal waste in plants?
Like, do you mean as fertilizer or like as happenstance?
Like as happenstance, so like landscaping that's meant for visual, and people taking their dogs and urinating or going on them.
I mean, in that type of situation, if it's like an urban area and there's going to be like a lot of dogs or a lot of cats present, I mean, the main risk to your plants would be like burn due to all the like ammonia in the pee. So that would be a problem. And like, if you've noticed that like vet clinics or dog parks, they'll often have they'll start out with trees, but they don't last very long because they get burned before they can get mature. And the same with bushes. So that would be an issue in terms of plant health. With people health, I mean, there are so many things to consider, but if it's just for like landscaping, I wouldn't worry too much about it. But if it's like a garden for consumables, I mean, really, the only thing you can do is just wash it thoroughly and use like a produce wash or soak it in like a vinegar solution, and that would help. But yeah, I mean, really, honestly, just physical barricading to keep animals out of like a food plot type planting would be the main thing. Does that answer your question?
Yeah, the burning. Thank you for that.
Yeah, because there's like so many diseases that they can spread to people. I mean, there's a lot that they can't, but there's a lot of infectious things that can be spread. And I mean, also even just like worms, not to be gross, but people can get tapeworms and roundworms and whipworms and all kinds of lovely parasites like that. So that would be my other concern, would be worms. But I mean, you've got to get like, yeah, like that's a whole different ballgame right there. So yeah, just prevention, and then making sure you wash everything before you eat it. But like for landscape stuff, there's not much you can do other than just like prevent them from getting there. And I don't think that there would be a ton of like health risks, you know, like for landscape stuff. So like for people working in it, I mean, obviously it would be best if they had like the little scooper shovel thing and just like removed it before they started working in the area, because, you know, just basic sanitary practice. So,
Gotcha. Thank
but
you.
actually, if you want to really pick someone's brain about that, Tammy Johnson, yeah, Johnson is her last name. I always want to put a T in there, but yeah, she is our wildlife infectious disease person, and she works on all kinds of things from mosquito-borne to tick-borne illness with animals and people and things that go back and forth between people and animals. And she used to work for the CDC, like on the animal zoonotic disease issue. So she would know more than I would.
Okay, thank you.
Yep.
I have a question about the mildew on things like squash and zucchini. It was very interesting to learn that they're species specific, that one plant won't have the same mildew as another plant. But I heard about a propagation practice just this year that many people trim off the leaves that are coated in the mildew as the vine grows and leave only the new growth at the end of the vine growing to help retard the spread of the spores of the mildew. Have you heard of that? Is it effective at all, or do you really need to rely on some kind of a fungicide to knock that down?
It definitely seems that it's like a sliding scale type of thing. It really depends on your inoculum density. If you have a very, what we would call low disease pressure, so little inoculum, and unfavorable conditions for it, then it would probably be adequate. But if you have a high inoculum amount starting out, like a really high population just from the get-go, when you start doing that, and also a really favorable environment for that pathogen, then I don't think it would be enough. So yeah, it's as unfortunate as that answer is, it is very case by case. So
Okay,
if it's like in a hoop house or something like that, that's a lot more predictable and easy to control. So, I mean, yeah, I could envision it being fairly effective there, where they can jack up the temperature and open up those fans and let it dry out really well. And then, you know, just stay on top of it before it becomes, you know, a high population of inoculum. So yeah, if it's in a controlled or semi-controlled environment, it's a lot easier to manipulate that with respect to diseases. So you could make it work in a situation like that more easily.
Thanks.